Corrector Tillman, whenever you're ready, we can start. Yep. Our prorector is experiencing some technical difficulties. We'll start in a couple minutes.
He's left a couple of messages to say he can't get his video to work. Tillman, correct.
Our prorector is experiencing difficulties, so on behalf of him, I will ask the candidate Hilaire Cheung to give his lay talk, and then we let Professor Tillman in again. Hilaire Cheung, the floor is yours for the next 15 minutes. Please unmute and share your screen.
Can everyone hear me? Dear prorector, dear members of the assessment committee, dear family and friends, I'm pleased to present the highlights of my thesis titled Deciphering Glycoprotein 6 Signaling in Platelet Activation, Integration of Functional and Computational Modeling Data in the next 10 to 15 minutes. First, for introduction, platelet plays an important role in hemostasis and thrombosis. Upon encountering damaged vasculature, platelet will first tether to the exposed extracellular matrix and then activate via a variety of receptor. One of them is the platelet collagen and fibrinogen receptor GP6. After that, platelet will undergo a series of events, including spreading, secretion of secondary mediators, integrin activation, and ultimately lead to thrombus formation. Due to the role of GP6 in platelet activation, can we find new inhibitors of GP6 signaling? The following schematic diagram show a brief summary of GP6 signaling in platelets. Upon activation of the GP6 receptor by endogenous ligand like collagen or fibrinogen, etc., it will undergo like dimerization and then activation and activates a series of signaling cascade through the FcR gamma, the ITAM, to the SAC, to the SIG, and then LED, and ultimately culminates in the activation of PI3K and PLC gamma 2. PI3K and PLC gamma 2 will convert the PIP2 into PIP3, IP3, and DAG. Among them, IP3 will also causes store mediated calcium release from the Yao store, and extra calcium will enter the platelet cell through the calcium entry channel. So the combination of the mobilized calcium ions and the DAG and PIP3 will ultimately lead to like platelet aggregation, secretion, and shape change. And therefore, the three highlighted being the GP6 receptor, the phosphoinositide kinases, and the calcium entry channel would be the potential targets for to inhibit GP6 signaling. Therefore, there are three aims in my thesis. First is to develop a mathematical model to predict phosphoinositide turnover in GP6-induced platelet activation. Second, to assess the effects of novel multivalent ligands of GP6 on platelet activation or inhibition. And third, identify the main calcium channel and carriers responsible for extracellular calcium entry in GP6-induced platelet activation. Moving on to the results. In Chapter 2, I've developed a novel mass spectrometric method for platelet phosphoinositide analysis. Platelets were isolated and treated, and then the phosphoinositides are extracted and analyzed with the developed mass spectrometric method. In the figure to the right, we can see the method is able to capture the CRP-induced phosphoinositides production, CRP-induced production or decrease after platelet activation. Afterwards, we have developed a mathematical model to capture phosphoinositide metabolism upon CRP activation in GP6-induced platelet activation. In brief, each species in the model is represented by mathematical equations. And for example, as seen here, the rate of change of phosphoinositide PI4P is equals to the rate constant of the conversion enzyme times the concentration of the PI. And these reactions would include a bunch of like phosphorylation, dephosphorylation, or hydrolysis, or transport across compartments. And afterwards, after the construction of the model, we are able to produce model simulations. And as seen in the simulations to the right, we can see the 10 best model simulations are able to fit quite well and converge with the experimental control data as shown in the previous slide with minor differences. Again, in Chapter 3, afterwards, we would like to try to examine the effect of different phosphoinositide kinase or phosphatase inhibitors on the whole metabolism. We've chosen PI4Ka, which is an enzyme which converts PI to PI4P and plays a role in PIP2 resynthesis. In experimental data, we are able to show that the PI4Ka inhibitors is able to suppress IP1 accumulation compared to control. Similarly, in model simulations, we're able to show the model simulations are also able to show the inhibition of IP1 accumulation, and in addition, also predicts the decrease in PI4P and PI45P2 compared to experimental control. Moving on to the second aim, we aim to assess the effects of novel multivalent ligands of GP6 on platelet activation or inhibition. In Chapter 5, based on a GP6 specific nanobody MB2, we've developed a dimer MB22, which is produced by linking to MB2 domain with a flexible linker. And as seen in the aggregation data to the right, we can see that MB22 is a more potent GP6 inhibitor than MB2, inhibiting CRP-induced aggregation at a lower concentration compared to the monomer. On the other hand, the tetramer MB24 is a platelet activator. As seen here in the aggregation curve, addition of MB24 induced platelet aggregation, which is in turn blocked by anti-GP6 Fab or the MB2 monomer itself. Moving on to the third aim, we've aimed to identify the main calcium channels and carriers responsible for extracellular calcium entry in GP6-induced platelet activation. In Chapter 6, we've used the following method to monitor calcium mobilization in platelets. Using fewer true-loaded platelets, we've added them to 96-well plate to test it in different conditions, and then add to an automatic dispenser and signal reader to monitor calcium mobilization in real time. As seen in this figure, we've tested a panel of we've tested CRP at different concentration and monitored the calcium mobilization with and without calcium entry. And under calcium entry, the calcium mobilization result in a higher amount of calcium mobilized. And in the calcium entry curve to the right, we can see an entry ratio of 5:7 for across all concentration. A similar experiment is done on thrombin, which is a PAR agonist, another major platelet agonist, and we can see it have a lower entry ratio of 3:4 compared to GP6. Next, we've used this method and test the contribution of different calcium entry channel on CRP-induced calcium mobilization. And among the inhibitors tested, we are able to observe that ORI1 is able to, and NCX inhibitor have lower calcium entry compared to the control. In the heat map below, where green indicates inhibition across the panel of inhibitor tested, again, the true ORI1 inhibitor and the NCX inhibitor results in reduced calcium mobilization in the presence of calcium entry, while the effect is less significant without calcium entry. To conclude, first, mathematical models can simulate the photoinositide metabolism of GP6 activated platelets and predict that the PIP2 synthesis is needed for sustaining GP6 signaling. Second, dimeric MV22 is a potent inhibitor for GP6, whereas the tetrameric MV24 acts as a potent GP6 agonist. Third, ORI1 and NCX have major contribution in calcium entry in GP6-induced platelet activation. And to sum up, inhibition of GP6 with MV22 or inhibition of phosphoinositide kinase or calcium channel proteins are potential antiplatelet targets. In the end, I would like to thank my supervisors, my colleagues and friends in Maastricht, in Birmingham, in Isastomon, and in Reading, and for the help and support throughout completing my PhD journey. And this is the end of my presentation, and thank you, and I'll give the time back to the prorector.
Well, thank you, dear candidate, for your clear and scholarly presentation. The opposition will now be opened by Professor Prinsen. He is Professor of Electromechanics of the Heart in the Department of Physiology of this university. He is also chair of the assessment committee of your thesis, and today extra secretary to the degree committee. Professor Prinsen, floor is yours.
Thank you, Mr. Prorector. Dear candidate, I have read your thesis with great pleasure. It's an extensive piece of work, well written, and most chapters also well published. And it shows elegantly how two universities can collaborate. But because the time for questioning is short, I save further compliments for later and like to ask you some questions now. In chapters 3 and 5, you describe mathematical models to simulate the processes involved in phosphoinositide metabolism. As you state in your last proposition, such models can be complementary to the traditional reductionist approach often applied in biomedical sciences, a statement that I wholeheartedly applaud. However, every model is a simplification of the reality. And I think it was Albert Einstein who stated that every model should be as simple as possible, but not simpler. And this is where I like to debate with you about. Your model utilizes mass action kinetics, but I think they do not include distances, location of certain molecules, for example, in the membrane versus in the cytosol or endoplasmic reticulum, or inner versus outer layer of the membrane, and diffusion distances. So I wonder, did you exclude them on purpose because they are not so relevant, or is this a next stage for your or other one's research?
Highly esteemed opponents, thank you for your question. I agree with what you have said, for sure, that every model is definitely not complete, and with as you've said, we've not include everything, and this is because of the simplification, because as you have said, like if a model is too complicated, like it can like create a lot of simulation that once like depends on how you swap the parameters, etc. And the the simplification we've chosen, I think, is of reason because like in the GP6 signaling, a lot of the protein and a lot of kinase or like PLC gamma 2 or everything are quite close and near in the similar area of patch near the plasma membrane set. A lot of the reaction are happening quite fast, but if we include like the diffusion distance or different area or more organelle, it could affect the result. And so like in chapter 3, for example, like there we do see like the GSKA1, which is PI4KA inhibitor, we can like in the IP1 result, we can see the inhibition, which the mathematical model is also able to predict. But on the other hand, the OCRL, which is a PI45, which dephosphorylated PI45P2 to PI4P, our model predicts something, but the data with calcium or the IP1 fails to show that. And we think the reason is could be like, as you've said, like the localization, whether the protein is transferred to the plasma membrane near where the active, where the biochemistry is, where the signaling is happening. That's what there's a mismatch. So I think there's something we can do and something we don't. So we have to know there's a limitation to this model, but we'll try to simplify and try to explain things within this, within the scope we've chosen.
Okay, and you tested your simulation, or you based your simulation parameters on measurements in test tubes. And to what extent do they also apply to the situation in vivo with streaming blood around?
Thank you for your question. I would say there are quite a lot of difference, like in test tube compared to in vivo. Like in test tube, we use watch platelets, and even like if we don't use watch platelet, we just use platelet-rich plasma. The behavior is already quite different, with like a lot more fibrinogen, a lot more like in plasma. There's a lot more thing which is not present in the watch platelet. And if you go to in vivo, there's even more like NO that is secreted by the vessels to suppress or PGI2, etc. So I would say it is limited to studying the watch platelets in a like, but for the in vivo, we need to incorporate a lot more parameter to consider more interactions.
All right, thank you for your answer. I give the word back to the prorector.
Thank you.
Thank you, Professor Prinsen. The opposition will now be continued by Dr. Jabari. She's reader in mathematical biology, School of Mathematics, University of Birmingham in the United Kingdom. And she's also a member of the assessment committee of your thesis. And please, all following opponents, take an extra minute to get in pace with the allotted time slots. Dr. Jabari.
Hi, Hilaire. So you've achieved something quite rare with your PhD thesis by using a combination of mathematical modelling and experimental work. Often you see one or the other, but rarely both in the same thesis. And I want to just drill down a little bit more on why a scientist would do that. I'd like to know what you think you learned from the modelling that you couldn't have learned with experiments alone. And that might be multiple different things because you've used modelling throughout your thesis.
Esteemed opponents, thank you for your question. So I would say it's really different, like in a mathematical mindset and a biochemist mindset. In mathematical, like everything is very quantified. Like in biochemistry, if just doing inhibitor, we're saying, oh, it inhibits and not, and there's a significant p-value. But then in mathematical, we have to look into like how many copy numbers is it in the protein, the enzyme, the lipid, the phosphorinoside, each one, like the starting condition really matters a lot, I would say. that give me a different scope. Like, for example, this is for the case for the phosphoinositide model. Even for the ABM model, it is also very different with different starting receptor, with different starting cross linker and phosphorylation rates. Like, they all give very different results. So that's what I learned to appreciate, how important the math, like the different scale would change everything a lot. So this is one thing. And another thing is that is how many species would just interact, like in the reductionist approach. Like as mentioned before, like we just test a lot of the time, we just test 1 or 2 inhibitors, and some, but not like 3 to 4 or 5. Then it would be a mess to do any control with. It's impossible in experiment, but it is something we can try and do in mathematical model, although it may not be accurate, but it's something that is possible at least. And also, thirdly, what I was trying to say, and also like what in biochemistry we always think, oh, a few time points, a few data with different inhibitors is important, but it is totally not enough in mathematical model. If you want to really good fitting and not overfit, you need a lot more model, a lot more data point than parameters. Like otherwise you will have overfitting very easily, which would not give a good result. So this is what I learned from doing both.
That feeds very nicely into my next question, actually. So whenever you're doing this cycle of modelling and experiments during your PhD thesis, at some point you have to stop. Are there further experiments you would have done to complement your mathematical models that you, if you'd had time, you would have liked to have done?
Thank you for your question. I will focus on the phosphoinositide model. In the thesis, I've just put in the calcium and the RP1 data because but for the best to really validate model, it would be best if I can show all the phosphoinositide data. But due to like COVID and also the lockdown and also like moving around, and also the mass spectrometry has to be done in a different institute, and then I couldn't really get the data by the time I published my thesis. So this is what I really want to add to show that the addition of GSK1 and the PI4K inhibitor would really inhibit the PI4P or PI4P2 as shown in the thesis or in the mathematical model or even in the slides. So that's what I would really want to be included.
And how about from a modelling perspective? Are there further directions that you would want to take the mathematical models?
Yes, so I think I've also mentioned some of the hypotheses, like things I want to include in the discussion. For example, like more components in a model, like the phosphatidic acid, like more, because there's a lot more in like IP3, IP1, like to CDPDAG, PI. There's a lot more behind, and so, but sometimes like I'm also not sure if it would work, because like sometimes we would include too much model without data, and it could also lead to overfitting. So probably for the best would be keep it this scale, because we don't have a lot of species monitored, like just 5 I've shown. Like if we over adding too much parameter, it could also like lead to like less accurate models.
Yeah, very good. And are there other modelling approaches that you would have, if you were to pass this project on to somebody else, would you say carry on with these ODE models, or would you say perhaps bring in other techniques as well?
There's I think of questions like, I think ODE, like at least for the phosphoinositide, it can explain quite a lot. But for the ABM modeling, for in chapter 5, I would also say, which is approach that works, then maybe it could be applied to the phosphoinositide model, but it needs so much testing work changing parameters, everything before it works. I saw my colleagues on the ABM model worked very hard to get it running. So it would be a whole another thesis than if you include another type of modeling, I would say.
I've got one final question if I've got time. Is that okay? So you talk throughout your thesis and in your propositions about mathematical modelling playing a really crucial role in drug development. What stages of drug development do you think modelling is really particularly useful for, or do you think it's throughout the whole course of drug development?
Thank you for your question. So I would say each part is related. Like, so this, what I'm doing is more drug discovery, etc. You have model to like simulate the effect of everything, to do some in silico, just some screening, etc. But like in drug development, like there's a lot of different things. Like I don't know much about clinical. Maybe I know more about the development in CDMO. Like for example, in upstream and downstream, like there's also a lot of mathematical modeling, just modeling the bioreactor, like the different metabolites or the cell growth viability, etc. is definitely a big thing. And also in downstream, in like how you optimize the parameter to purify with least impurities is also huge. And for clinical, maybe with more like, I don't know, more data, a lot. I don't know they're doing a lot of NGS, like genomic data sequencing, to get the phenotype of different patients and to sort of predict what could happen. Like, can you like minimize safety and to choose the right dose, etc. I mean, maximize the safety, minimize side effects, and choose the right dose, etc. So it's in every aspect, I would say. So it's just how you use it and how you approach the problem.
Thank you very much. I'll hand you back to the prorector now.
Well, thank you, Dr. Jabari. The opposition will now be continued by Dr. Hawkins. He's a group leader in cell and molecular biology at the Birmingham Institute and University of Cambridge, United Kingdom. And he's also a member of the assessment committee of your thesis. Dr. Hawkins.
Thank you, Pro-Rector. So, dear candidate, can I start by asking you to read out your proposition 1?
Thank you for your question. So my proposition 1 states that unchromatography coupled mass spectrometry allow to separate the positional isomers of phosphorinositide in platelets, particularly PTDINS 34P2 and PTDINS 45P2.
Thank you. So first, I would like to congratulate you on the progress you've made developing this new methodology. But I was interested in seeing that when you applied this method to measuring platelet phosphoinositides from different donors, that the variation in technical replicates from the platelets from a single donor were really nice and tight, showing your method works very well. But you got quite large variation between biological replicates, i.e., platelets from different donors. Did you ever try and correlate those changes with any other parameter of the platelets from the different donors? For example, their ability to aggregate or their calcium responses to different agonists?
Thank you for your questions. So yes, indeed, like so as you've mentioned, in the data, especially like the PIP3, we see quite a lot of discrepancy, like especially among the biological replicates. And at the time of preparing the samples, like it was done in Maastricht in 2019 September. In addition to preparing the phosphorinositides sample, I also did the measuring the calcium response, calcium mobilization response for the upon CRP or thrombin activation. And I did see a change, like because why there's a big difference I attribute is because some donor have much faster response, spike earlier PIP3, maybe in the first 15 or 30 minutes, and some have a slower response, like at like 120 seconds, etc. At least with my data. And similarly, in the calcium traces, the donor with faster response do give higher spike and quickest and higher and quicker spike in calcium mobilization compared to the donor, which gives slower response on PIP3 and calcium.
So can I stop you there and ask a follow-up question? So given that you're seeing these differences in platelet behavior, did you think about building your mathematical models for each individual donor? So get some really good high number of technical replicates from a single donor, build your model there for that donor, then compare it with a model built on separate data from another donor, and then see if it can help explain these changes in sensitivity, speed of responses, etc.
Thank you for your question. When I think back, back to your question, when I think back, like what is happening, why some donor have faster response, some donor is slower response. It could be like different GP6 copy number or different downstream like kinases copy number in each donor, and I think these are like quite this is like included in part of the STEAM function in the model, in the model phospholinositide model. So we are just giving us one curve we're just doing the average for now.
But what I'm asking you is, did you think about building a different model for each donor to see how different they are?
Yeah, I see what you mean. So if you have different model so I think there's two ways to approach this. The first thing is to keep the parameter the same with all the like the species with all the species and reaction enzyme parameter or species copy number, keep it the same and just change the STEAM equation based on the different donor and based on the downstream signaling all the way to PI3K and PIC gamma 2, and see if the same parameter can give different responses accordingly. This is one approach. Another approach would be don't change the STEAM function and then change each reaction parameter based on each donor, but then this would lead to a very complicated model. Like you have different parameters for each patient, and you have to
that could be very but that could be very revealing as well.
Yeah.
Okay, thank you for your answer.
I would say you need to like do both approach and see which is the better to give a better result. But
I agree. I will hand you back now to the pro-rector. Thank you.
Thank you, Dr. Hawkins. The opposition will now be continued by Professor Smith. He's a chair of cellular physiology of the Department of Molecular Cell Biology, University of Leicester in the United Kingdom, and also a member of the assessment committee of your thesis. Professor Smith.
Thank you. Can I just say well done on writing an incredibly well written and understandable thesis. I really enjoyed reading it, and I learned a lot. I mean, we're not an expertise in all areas, so you know, it was really illuminating to read many sections. I guess I have one general short question and then some more detailed questions about chapter 6. Just a general question, slightly outside of my area of expertise, so I hope I phrase it correctly. The clustering of GP6. I mean, can you explain the relationship between that and this dimerization versus tetramerization that are required for activation of the receptor? So in other words, what does clustering mean for activation of that receptor?
Highly esteemed opponents, thank you for your questions. So the way GP6 activates have been studied throughout many years, and our understanding, I think, also changes a lot for GP6. And for dimerization, I think it's the two GP6 molecules can be dimerized through the extracellular domain through the interaction. While for the clustering and the activation, so like we have a lot of so GP6 is constantly each GP6 is constantly bind to like the FCL gamma, and FCL gamma is also activated activated by SAC, and then which activates SIC to, and then which gets to downstream signaling. But then at the same time, there's also regulatory effects. So there's a phosphorylation pressure like by the kinases, but also dephosphorylation pressure, for example, by SHP1 or 2, like something that keep dephosphorylating those kinases to prevent its activation. So clustering is that like upon, like, for example, in collagen, like in the ECM on the surface, so there's a high density of the GP6 epitope on the surface, and then this GP6 would be able to bind at the on platelets will be approaching those epitope, and then are close to each other. With that, there is a much closer, much more high density of the, for example, SAC, because it's constitutively near the FCL gamma, and then with the high increased phosphorylation, while the dephosphorylation pressure is like less significant, there's a accumulation of phosphorylation which ultimately lead to a lot of the SAC or SIC phosphorylation and all the downstream signaling. And yes, so briefly speaking.
Okay, I mean, I reflect on, I understand GPCR signaling more than I do tyrosine kinase signaling, and there's this concept of cooperativity, and I just leave you with that thought, with that general question as to whether there is any overlap in that concept. But on then to just one or two questions about chapter 6. I mean, in Table 3, page 228, I'm not sure if you can quickly turn to that. You have some numbers there that very nicely summarize the effects of the small molecule inhibitors, and they're all normalized, I think, to a specific condition. But I was what struck me was the quite substantial effect that those SMIs have on the collagen-evoked calcium response compared to the thrombin-evoked calcium response. And I just wonder whether you can elucidate on that. Have you got any additional thoughts and explanations for that?
Yes, thank you for a question. So you've mentioned like the SMI effects on collagen-induced calcium mobilization, right?
Yes, the ligand you're using is CRP, isn't it?
Oh, CRP, not collagen. Okay. So
you also have thapsigargin in there, so you're I guess stimulating everything that you can.
Stimulating. Oh, it's with thapsigargin.
Yeah, thapsigargin alongside CRP, or thapsigargin alongside thrombin.
Yeah. So yes, I am here in this data. It shows that CRP in the presence of calcium entry, and also thapsigargin leads to a very strong calcium entry, like up to 10,000, 20,000, etc. While thrombin is less significant, it's like 10 times less, like 2,000, etc. This is observed in this study, which is quite interesting. But it is also sort of like showed somehow in some previous study, for example, in some ORI1 knockout mice, they show that like is the whole stem aura is more coupled to the CRP induced calcium mobilization compared to thrombin. Like in the mice, like the CRP induced collagen induced calcium mobilization is much more inhibited in the presence of extracellular calcium compared to thrombin in that study. So it's sort of like, yeah, correlate to each other, but like you can't say for sure because like one is mice, one is human platelets, but they do have some previous studies showing that the more coupling of stimuli with this collagen CRP.
It did make me wonder under what conditions the platelets are in when you hit them that hard with stimuli, and whether the thrombin on top of particularly quite high concentrations of thrombin are leading to some sort of loss of membrane integrity. Did you ever look at the platelets afterwards in the wells to see what they will look like?
We did look at it, but we didn't really look through the microscope. We after we take out the plate, a lot of time it's just aggregated platelets, like or white dots in the middle, and yes, but not really look into a microscope and see the integrity of the membrane, etc. Yeah.
Okay, thank you. I think I'll stop there because of time. Hand you back to Pro-Rector.
Well, thank you, Professor Smith. The opposition will now be continued by Dr. Baten. She's researcher in hemostasis and thrombosis at the Department of Biochemistry of this university, and she's also a member of the assessment committee of your thesis. Dr. Baten, please go ahead.
Thank you, Mr. Pro-Rector. Dear candidates, first of all, I would like to congratulate you with the nice work you have done and presented in your thesis, and also thank you for the nice overview of your most important findings. You gave a very clear presentation. Of course, I would also like to extend my congratulations to the promotion team. I would like to discuss chapter 4 with you. I'm also very interested in the concept of reversal of platelet activation, and I would like to discuss your results with you in some more detail. But first, you could clarify something. You could perhaps clarify something for me. Throughout the chapter, you refer to reversal of platelet activation and platelet deaggregation. But when I read the last paragraph of your discussion, it left me a bit confused because you mentioned that strong aggregation appears to be an irreversible process, which is also reflected by your findings. But then you conclude that the small reduction in aggregation upon SARC inhibition is likely to reflect loss of contraction on page 139. So my first question to you, the increase in light transmission we can see in your experiments and that you observed upon SARC inhibition, is that platelet reversal, disaggregation, or is that more loss of contraction?
Esteemed opponents, thank you for your question. So I would say it can be so. Yes, there reduction in aggregation measured by LTA in the presence of SARC kinase inhibitors. So because like in the integrin, outside in signaling is also linked to the actin filaments, etc. And previous study have also linked like the integrin outside in would be related to clot retraction, etc. So this is possible, but like it for sure happens in a longer time scale, not like just 300 seconds after I add the agonist. So probably like what you have said could also explain the
If I understand you correctly, you say it's a combination of both. So both platelet deaggregation, so loss of platelets, single platelets from the aggregate, but also a loss of contraction. Is that what you're saying?
Yes, could be both. Yeah.
Okay. You also mentioned in the discussion that SARC and SIC inhibitors can promote deaggregation under flow. So from deaggregation, release of single platelets from a thrombus. in your setup, you find very limited disaggregation. There is some disaggregation, but the effect is rather small. So how would you place your findings then in the findings you refer to from literature?
Thank you for your question. Yes, in this finding under the aggregometer setting, I do not really see a lot of deaggregation with the SAC or SIG inhibitors, while in like previous study with flow chambers in Maastricht in Birmingham, we did a lot of also like coating collagen and then run through the whole blood and see, and then afterwards adding kinase inhibitors, and we see a much more profound disaggregation compared to that as seen here. So I would say the disaggregation really depends on the presence of the shear force, for example, in the flow chamber, present in flow chamber, and also like the strength and the cost of activation pieces in the flow chamber, like the platelets are only the collagen are only present on the slides, but in the LTA is in solution in the CLP. So I would say it's a different settings, and with different settings would come different results.
Mm-hmm. I agree with you on that. Would the methodology you used also affect the the rate of disaggregation you detect? So in other words, the LTA assay, is it sensitive enough to pick up those very initial traces of disaggregation, or is that easier done when you have a flow chamber setup where you have the combination with microscopy? Could that also be of influence?
Thank you for a question. So I think LTA can also be used to measure like disaggregation, like previous study with ATP as agonist did with similar experimental setting, also in an aggregometer with ADP and then add eptifibatide. The study did observe full disaggregation, unlike this study. So I would say, like, in addition to this, while for GP6 induced activation, like we don't see at all in this study, at least. So in addition to the experimental setting, so the choice of agonist also matters, like so.
And so this is of course in vitro. How would you place this process of platelets deaggregation, reversal of platelet activation in a more in vivo setting? There you of course have also coagulation, so there will be fibrin that will stabilize the clot. So in the process of thrombus formation, when would this platelet reversal of platelet activation, when would this occur, and when would it be important?
Thank you for your question. So in vivo, like with the much more, like as you've said, there's a coagulation happening at the same time with platelet activation as seen here. So in the study here we showed like the reversal of the tyrosine kinase phosphorylation, but we already don't see the platelets disaggregation at all. So with in vivo, I would imagine like with the addition of fibrin and also a much more concentration of fibrin, and everything goes much faster. I would say it would even be less, like, because everything would even goes on faster, and like compared to here in this study here.
Would that then also be would the location of the platelet within the thrombus then also be of importance there, when you also on top of that have coagulation ongoing?
Yes, the platelet location definitely matters, like, but it depends on what is the cause of the thrombus. Would it be like exposed vasculature, or would it be a atherosclerotic plaque where it's already quite full of collagen, etc. So depends on different setting and where what's the agonism, where can they be found, etc.
Yeah. Okay, thank you for your answers, and I would like to give the word back to the prorector.
Well, thank you, Dr. Baten. And the opposition will now be actually closed by Professor Owen. He's Chair of Immunology and Immunotherapy of the College of Medical and Dental Sciences, University of Birmingham, United Kingdom. And he's also a member of the assessment committee of your thesis. Professor Owen.
Thank you. Thank you, guys. It's good to see you again, by the way. I hope you're doing well. So, I really like the thesis. It's kind of quite rare to have people be able to do the kind of practical work and the modeling work together like that. There aren't many of you around with those kind of skills, so I really enjoyed it. I especially enjoyed chapter 5, the kind of modeling and the multivalent ligand work, because, and I guess that we try and do similar modeling in my lab on T cells and things like that. So that's what I'd like to kind of explore a little bit with you. I guess what you start off by doing is looking at kind of artificial ligands, which are monomers and dimers, and then you move to tetramers, and you see the kind of response of those. I was wondering what would happen if you look at the other oligomers that you could do. I mean, you kind of skipped trimers, so it would be interesting to know what would happen with trimers. Is there something about, they have to be in pairs, or would a trimer work? And what about higher level oligomers?
Highly esteemed opponents, thank you for your question. So I would say first for trimer, like MB2, we know that it will also activate platelets based on the data finding in Birmingham platelet group. So it does activate. And for higher oligomer, then I would say imagine it would be like CLP or collagen, because they are the GPO is and the GP6 are like lower affinity compared to the nanobody or specific antibody, but they still show like activation, like even if you have lower affinity. So yes.
In terms of doing those, with modeling, you can kind of model anything you want. But with the wet lab experiments in platelets, what would be the practical limitations to making higher oligomers of the ligands? Could you feasibly make a hexamer, let's say?
So if you are saying like if the collagen related peptide where it can be synthesized, they just put 6 more GPO motif together, but I think without cross-linking, it's not going to activate anything. While for antibody, like for nanobody, at least for me, like I would say that it's very difficult, because compared to a dimer, the tetramer already have a much lower yield production by the hex cells compared to dimer. I've done it many liters, many liters of bacteria, but it's not increasing a lot. So that's the risk. Like the larger is the protein of the nanobody, more you cross-link like this, it's harder to have high yield. So if you don't have enough reagent, then it's hard to do an experiment.
So just thinking about the kind of cross-linkers rather than the ligands, so when you're kind of pre-clustering the receptors. We have a project in our lab where some company wants to test multimeric antibodies that can kind of pre-cluster receptors. What do you think? It'd be good to get your thoughts in general on what are the kind of therapeutic potential of drugs which alter, let's say, nanoscale clustering of receptors. Do you think they have potential in the pharmaceutical sector?
Thank you for your question. I would say it would be quite hard to characterize the exact binding and way of mechanism or how you prove its activity, because like in at least with MAB or bispecific or similar reagent, like it's everything, the activity is very well characterized, the mechanism of action, and you have to have the exact mode of action, but with cross-linking reagent. I'm not sure the exact way of doing it, but for example, if it's a chemical biological probe or di-cross-linker, like quick, and then inside the cell, then sometimes it's hard to control the way it cross-link, like different amine or SH group, and they can have different interaction. They can 2, 3, 4, or 5. They can face together or face opposite. But so it's doable, but I would say it would take a lot of study to characterize the mode of action, and also like how you optimize the agent to give you the best response, how you make sure everything is facing down towards the receptor but not going up and down. That wastes a lot of reagent and have some potential crosstalk with other cell type, which would have some unintended side effect, etc. So there's a lot of design work that would be needed to make sure it works as intended.
Okay. I don't know, it's kind of a bit of a difficult question, a bit of a general question, but obviously in some signaling pathways, the ligand is monomeric, in others it's dimeric or higher oligomers. Do you know why that is? From an evolutionary perspective, why some signaling pathway or receptor would prefer a dimeric receptor compared to a monomeric one?
Thank you for your questions. So evolutionary.
I don't know the answer either.
So just say for GP6. So GP6 intended ligand like fibrinogen or fibrinogen or collagen, right? So they are all like long length and have many epitope, etc. So maybe you want a receptor that is especially GP6, which can produce a strong and sustained signaling. You want them to activate as specific as possible, so that only if there's a large area of the ligand, and then it can bind on, so that it can activate. Otherwise, if it has other high affinity soluble ligand in the body, then it could activate easily, which give unintended thrombosis. So maybe it's some design. And while on the other hand, like other thing would be like, so, but like for other receptor, other tyrosine kinase link receptor, like EGFR, for example, being a dimer is in the mechanism. You have to dimerize to actually unfold the kinase domain to actually signal downstream. So depends receptor by receptor.
Okay.
Yeah.
Just thinking about your data for a second, and obviously your PDEs were kind of producing these bell-shaped kind of dose response curves, your concentration curves, which I think were not really recapitulated then, in practice. So to what extent, and I think you give a brief explanation for that in the thesis, but to what extent do you think that kind of illustrates a limitation of PDEs for kind of for modeling these kind of phenomena if they don't really recapitulate, what you might get out of an agent-based model or what you might get out of an experiment?
Yeah, thank you for your question. So I would say, well, ODE like PDEs are definitely used a lot in pharmacological question, which explain a lot of phenomenon, definitely. But then like sometimes like for different receptor, for example, in this case, GP6, you have like low affinity and you also need clustering that you may need to add other parameters to actually encapsulate the whole receptor ligand interaction, and also like anything happening below the in intracellular, not just extracellular, because it also affects. So to actually give a more accurate representation, it's not it's like we need to add more instead of just using maybe just a L plus R equals LR. Yeah, I think we need more parameters probably to better encapsulate it.
I think we're just running out of time, but can you briefly maybe just give what would be the one major thing that you'd incorporate into the ODE model in order for it to better recapitulate the
Um,
experimental data?
Ora asked.
Dear candidate, dear Hilaire Cheung, the time appointed for defending your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and your defense. I request that you and your company await the results of our deliberations and our return. distinction in accordance with Dutch university custom. I invite your superior now to take the floor.
Dear candidate, we first get the promise. Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
Yes, I promise.
Okay. Then, by the authority vested in us by law and in conformity with the decision of the committee here present online, I hereby confer upon you, Yam Fung Hilaire Cheung, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, you will soon receive the degree certificate signed by the rector, the secretary, and the supervisor affixed with the official seal of the university shown by the beadle. So, dear Hilaire, dear Dr. Cheung, so now it looks a bit strange online, but now you have made it, you now are a doctor. So my sincere congratulations for obtaining the double degree of the universities of Birmingham and Maastricht. And in my congratulations, I also involve your family and friends who hopefully are able to follow this ceremony online. Since you are in a distance, my advice for today is make sure you celebrate this degree in the best possible way. In laudatio like this, it's common to look back the past 4.5 years and see how you made it from an excellent master student in Hong Kong back in 2018 to double doctor today. And myself, I will confine to some parts of the story, and later on Professor Steve Watson will more concentrate on the longer time that you spent in Birmingham. Your CV in 2018 indicated that you had already experience with double studies. In 2016, you followed a double master in chemistry and biochemistry at the University of Hong Kong. And this combination of biochemistry and chemistry, together with your relevant work experience, including mass spectrometry, was the reason why we selected you as an early stage researcher, abbreviated as ESR. So you were incorporated in one of the projects of the TAPAS program. It was financed within the Horizon 2020 framework of the European Union and led by Professor Steve Watson and Dr. Nathalie Poulter. From the very first start, I remember that we were unsure about your age, because only in 2019 there came an adapted CV with your birthday on. TAPAS stands for Targeting Platelet Adhesion Receptors in Thrombosis. And as you can read in your thesis, this is exactly what you have done in a quite successful way. But looking back in the TAPAS application, it took me a while to find your project because it was quite general. It was entitled Integrating Omics, Functional Data Sets and Microscopy to Predict Thrombosis and Hemostasis. Let's say that this was a quite broad project title. Broadly speaking, it was a hardly impossible project, and very strictly speaking, this is exactly what you did regarding platelet, GP6 and modeling work for the thesis. You were appointed at the Leibniz Institute for Analytical Sciences, ISAS in Dortmund, and started there under supervision of Dr. Robert Arendt and Professor Albert Siegmann on a novel lipidomics project on identifying the phospholipids, phosphoinositide phospholipids in platelets. And this combined chemical and biochemical research has formed the basis of the present thesis. Later on, you worked with groups in Berlin, Alagras, Theranostics, Reading, Maastricht, and Birmingham. And here you collected an impressive number of supervisors. I only mention a few: Dr. Bote Lange, Dr. Christoph Wierling, Dr. Joan Dunster, and Dr. Marijke Kuipers. So rewording your TAPAS project, you made it to integrating supervisors, institutes, and ideas to predict platelet functions in thrombosis and hemostasis. Reading through your thesis, and this is also the opinion of the opponents present here today, this integration worked quite well. Here in Maastricht, in my group, you've made a terrific effort to determine the major calcium entry channels in platelets, and furthermore to use calcium measurements for the validations of your phosphoinositide modeling, such as you explained in the late presentation earlier this afternoon. Now, before handing over the microphone to your first promoter, Professor Steve Watson, on behalf of the promotion team, I'd like to thank here all the members of the assessment committee, Professor Fitz Prince and Dr. Constant Bate. Thank you for presenting the expertise from the University of Maastricht, Dr. Sarah Jabari and Professor Dylan Owen from the University of Birmingham. We really appreciate your willingness to judge on the extra thesis defense here in Maastricht. Dr. Philip Hawkins from the Birmingham Institute and the University of Cambridge, thank you for bringing in your quite valuable experience in phospholipid research. And last but not least, Professor Martin Maat-Smit from the University of Leicester. Thank you also for bringing in this valuable expertise. Now I hand over to Professor Steve Watson.
Thank you very much, Dr. Heemskerk. Hilaire, many congratulations. I think I'm going to echo much of what Johan has said, and I'll take this opportunity to in particular thank the opponents before I say a few words about your own reflections on my own reflections on the last few years. I think that they gave you a very fair and challenging series of questions, and I think you gave a very clear presentation. I think some of your answers were really excellent, and I think one or two of them I think have given you more to think about. And I always say, if you get a good set of opponents, you'll learn a lot about the thesis from the questions that are asked, and I think you can take some of that on board. I think to have two defenses of a PhD is something that I don't think anybody else in this Zoom meeting would like to have gone through, and I can assure you that you're never going to have to go through this again. So really many congratulations on that part. I think Johan has already described the challenges of the project from the outset. Perhaps I'm going to give a little bit more deeper understanding of the challenges. You started in ICES, a fantastic place to do research, but it's a non-academic institution, so I was not able to confer degrees. And this is why you had three effective moves over the course of the 3 years. Your first supervisor, Robert Ahrens, your day-to-day supervisor, left before you left ISAS. And although you were able to develop a fantastic method, you were not able to really fully exploit it over the 3 years, partly because of the pandemic and other factors. And I know that some of that work has only recently been complete. And I think you made that point in one of your answers. You arrived then in Birmingham in February 2020. In March, Lewis Moran, another student on the TAPAS program, arrived in Birmingham, this time from Santiago. 2 weeks later, the university shut down for the next 4 months. One of my lasting memories of that time was our weekly Zoom meetings. I know that you'll never forget them. I was probably your only serious contact with the outside world. I think you might have gone and got a supermarket shop once a week, if you can remember then. But outside your room, it was that room, and it was always that wardrobe behind you, and it was that suitcase at the top that I still want to know whatever was in that suitcase. And I came up with a whole variety of theories. It's challenging for a supervisor to come up with an experimental arm of a thesis when you're not in the laboratory. And I think I need to a fantastic thanks to Dr. Jo Dunster. I hope that she's on the call, in the University of Reading, who stepped up to really support you on the mathematical modeling. I think your thesis, you are unusual in having a strong concept in so many different areas, but I think you will not describe yourself as a modeler, but I can assure you you are much more than most cell biologists. But Joe was really very instrumental to that period, and I enjoyed our discussions around the science. We also wrote you contributed to a couple of reviews, but that was a challenging time. We opened up in July. This then brought an even further challenge because we opened up in bubbles. The lab started from 9 till 12:30, 9 till 1, 9 till 12:30, and then 9 till 1, 1:30 till 5 o'clock. And so as a consequence, one person was in one group, was in one bubble, another was in another bubble, and the experiments took 6 hours. So Lewis would prepare the platelets, he would not be able to meet you, you would then do the experiment, vice versa. And this is actually partly how the aggregate stability chapter came along, because we had to have something relevant to both theses, and it was really quite a challenging time, especially as you were not experienced in making platelets and doing the work at that time. I think it's testament to how you managed to get on and achieve so much. We actually only really came out of lockdown at the Omicron when it was 200,000 cases a day at the end of January, early February, by which time we were already making plans to move, for you to move to Maastricht. But one of the things that came out of the lockdown, and there were some benefits, was the time to think more. And having made the nanobodies, we then considered, could we cross-link them as something to actually produce the first ever defined agonist, not just of GP6, of CLEC2, and we've since rolled this out to other receptors. I think it's Elena Martin who really came up with the idea in conversation with myself, but then we then had to get them expressed and so on and so on, and that project has continued on. And you were able to model this, and again we mentioned Christoph Verling and Alakris and Zara Massoud, another TAPAS student who you also worked with. I think what you've shown yourself over this period is the extent to which you are a team player, to which you are a great thinker, and to which you have got enormous flexibility. I don't think that everybody in Birmingham really got to know the true Hilaire, because you were only actually properly in the lab for a relatively short period and on a day-to-day basis, and the lab was still adjusting to coming back. But what I think we did see was a very talented and group team player who I think has got an enormous potential as a researcher. You've chosen to go into industry, and I know that you will do extremely well there. I think we didn't get the full academic curiosity out of you that I know you have, because it takes time. And I think but I know you have that there. And I think if you ever wish to return to an academic environment, I'm sure that every one of us would see that you have a tremendous potential. And you've got the advantage that you're young enough to decide what you want to do in that way. So many congratulations again. I hope your family are online, and I hope that they should feel very proud of you. I know as supervisors, we feel very proud of you indeed. And we wish you very well, and we ask you to stay in touch. And with that, I believe I'm handing back to the pro-rector. I just want to thank the opponents again and in that way, and University of Maastricht for organising this defence. Well done.
Well, thank you very much. And dear Dr. Cheung, also on behalf of Maastricht University, I congratulate you with the degree that you have now acquired. And in this congratulations, I would also like to include all your family members, friends and colleagues that have joined today's marvelous ceremony. From the Today Show, I can really appreciate that you are a much appreciated scientific team member, and that is really good to hear. And thank you also for these nice words in the Laudatio. And also the committee members, I would like to thank them for their oppositions and of course for their flexibility. And with that, I declare this ceremony ended.